Okay, hello everybody, and I know we had a little switcheroo. That's the only switcheroo I'm really doing is like, I figure like, on Wednesday, the digestive system, especially like simple physical and chemical digestion, that set of digestive system stuff is pretty easy, so that's why I put it on Wednesday. I really wanted to talk about immunology on Friday instead of having the rest of the immunology just be pre-recorded because it's a pretty complex topic and it's a really very topical and relevant topic today. And it's very interesting, a little bit of like recent history. I mean, about a year ago, if you somehow were here last year, we still didn't even have exam 2 yet because it was like we didn't know how to have the exam. We still needed to make preparations. We came back from spring break and everyone was moved online. We kind of prepared for moving online before, but most of pretty much the entire university that wasn't prepared found out that we had to move online during spring break. So we had a week to get our stuff together. So it's interesting to go back to the same this around the same time last year. I talked about we didn't have a vaccine. We didn't know what could be we didn't have any treatments for coronavirus. All we could do is like do our best to keep the people who were infected alive, but there was no vaccine. A year later, now we finally have vaccines, so it's very interesting to cover what happened. Okay, so hopefully you saw the lectures on Monday because again, the if the adaptive immune system and all the cells seem very, very complex, that's because they are. And again, this is where you should keep it limited to what I cover in lecture because there is a lot of information about this and the rabbit hole goes down even deeper. So pretty much what I cover in lecture in terms of immunology is pretty much what I test on for this class because again, there's so much about this. All right, so here we have a simple pathogen invading the body. Neutrophils destroy the pathogen. Now you have all these pathogen particles. Remember dendritic cells. So dendron means some sort of tree and branch. And what they have are many branches, and they send out their little feelers, they sample the surroundings, and they try to find any of these particles and antigens in the surroundings. And what they do is sample the surroundings, and then they go to the lymph nodes, where you have all these naive T cells. And what happens is that these dendritic cells, they start to presenting the antigens they found in the environment at the lymph node. And this is why, remember my analogy, so these are naive T cells. And again, naive does not mean stupid, it just means like they haven't been educated, they haven't been learned yet. This is why it's like that. So the thing is that naive T cells are like brilliant cells, but they haven't reached their full potential. That's why professional antigen presenting cells, I use that analogy, they're like professors. They kind of teach naive T cells who are budding professionals to become fully developed professionals. So these are naive T cells and they're going to be taught, and if they recognize these antigens, then they can graduate to become professionals. Now, or I shouldn't say professionals, they become fully developed toward their career. So if they have the appropriate T cell receptor, and for some reason T cell receptors aren't showing up, but if they have the correct T cell receptor and it's a match, then these naive T cells can be activated and become either cytotoxic T cells, helper T cells, memory T cells. There are many flavors of T cells, but they're all going to come. If you have that T cell that has a receptor against one of the antigens presented by a dendritic cell, the T cell is going to try all the T cells that have that same receptor. They're going to try to fight anything that has that antigen. Now, here we have a cytotoxic T cell. And the interesting thing is that when a dendritic cell activates a cytotoxic T cell, well, it undergoes replication. So these cytotoxic T cells are going to make multiple clones of themselves. So they all have the same, they're all going to recognize the same antigen here. So they all have the same like T cell receptors. So these are going to proliferate, they're going to divide, they're going to go under multiple rounds of mitosis until you have many of these types of cells that again recognize the same antigen. So collectively, this is what we call a clone because why? Again, they're undergoing mitosis and recognizing the same T cell. And these are effector T cells because they're actually doing the immune system function. They're actually trying to combat and fight against this pathogen that has the antigen they're recognizing. Now, the thing is that not all of them become active. The interesting thing is that even though this cell has the same T cell receptor, it's actually going to hold back. It's not going to divide, it's not going to undergo mitosis, and this is what we call a memory T cell. So it still has the same recognition of the same antigen, but it's not heading off to combat. So it's kind of like a little reserve forces for your immune system. Now, the effector T cells are going to fight against the pathogen and die during the process. Or when you clear the pathogen, these effector T cells are no longer needed, so their body gets rid of them because it takes a lot of energy and nutrients to maintain cells. So if your body doesn't need the effector T cells, it'll let them go. Now, these memory T cells, they don't die off like the effector T cells. They hang around. But again, you still have these T cell receptors that recognize that original antigen presented in that initial infection. So these memory T cells, they remain dormant. They're not going to divide, they're not going to become effector T cells because now you've cleared the pathogen and you're done with that for now. But what if you get reinfected with that pathogen? And then you have the antigens from that pathogen entering your system again. What is that going to do to your memory T cells? It's going to activate them, and now they're going to divide. So these memory T cells are going to proliferate, undergo mitosis, and still create some memory T cells as well, hopefully. And then these will become effector T cells. And this is why when you have a second exposure to an antigen, why it's a faster response if you have these memory T cells. Because you don't have to go through those multiple rounds of dendritic cells grabbing antigens, trying to train new naive T cells, trying to generate more memory cells and find a match with the T cell receptor. When you already have a T cell receptor and this population of memory T cells that recognize the antigen, they can just hit the antigen again. If the antigen hits these memory T cells, they can just explode in population, and these can go, effector T cells can go off to war and fight against that second wave of the pathogen. So again, the main point from the previous slides is that not all T and B cells become active. Some of them hang back and are stored for later, so they don't go off to war. They kind of just hang out. So memory T and B cells, you can have both memory T cells and memory B cells. And again, they're kind of like records of antigens. So they kind of have, this is why they're called memory T and B cells. They're hang around and they're records of what you were previously exposed to. And again, because you don't have to do the training of T and B cells again, because these are from the original exposure to the antigen, this is why the subsequent response or exposures to the same antigen, the response is faster, because again, these cells are already trained. They already recognize the antigen. And then, so again, it's kind of like a reserve forces. So again, the effector T cells are the ones that are like active combat, but these are kind of memory T and B cells are kind of like the ones that are in reserve, so you don't spend all your forces. Because if all your cells become effector T cells, then once the infection is clear, you have no record of the previous antigen. Okay, so why is this important? Well, this is basically memory T and B cells are the principles, are the underlying physiology and principle behind vaccination. What you're trying to do with vaccination is that instead of trying to prepare your immune system ahead of time, instead of relying on infection to train your immune system. So what you do is take the antigen from some sort of pathogen. It could be a bacteria, it could be a virus, and then put it into a vaccine, and then expose your immune system to antigens from that pathogen, and then try to train your dendritic cells, your professional APCs, your B cells, your T cells, and try to build this reserve of T and B cell, memory B and T cells before you're actually exposed to the full pathogen. So yeah, what we have here is, now this is actually an old slide. It says like live virus vaccine, so you can have a weakened virus or you can have an inactivated virus vaccine. So you can take the virus itself and somehow chemically process it, so it's either not able to replicate as much, or it can't replicate at all. Or you can get the DNA that has like proteins from the virus. Now we have that new mRNA vaccine. So that mRNA vaccine, the technology wasn't new, but this was the first time with like Pfizer and BioNTech and Moderna This is the first time it's actually being used in humans. So the technology has always been there, but proof of concept and actually carrying out to human trials, that's a new revolution. So I should have added mRNA vaccine too. So there's multiple routes now.